Adaptive study designs are defined as any multi-stage study design that uses accumulating data to decide how to modify aspects of the study without undermining the validity and integrity of the trial. This means you're going to enroll some subjects, look at their responses, and then make changes to the study, like increase the sample size, or stop randomizing to a treatment arm, or stop the study entirely. What you examine and change in the study defines what kind of adaptive design you're doing. As you can imagine, clinical trials are really complicated things, so that can cover a lot of ground. The clinical trials world has therefore settled on a few key designs: adaptive randomizations, sample size recalculations, Bayesian dose escalations, and group sequential studies. Now, the group sequential contingent includes seamless phase 2/3s as well as pruning designs, both of which are what people usually think of when they talk about adaptive designs. But don't be fooled. The others are perfectly legitimate, useful designs. They're just not as hip and trendy. So each of these examines the result of one piece of the study as it progresses and then makes changes based on what's observed. That's the key. The reason the group sequential type studies have gotten so much press is that these types of analyses look at outcomes in larger studies. So there's more potential for changes and theoretically more savings. The underlying concept behind the group sequential studies is that you have multiple interim analyses, but you still control the type 1 error rate. The savings happens because on average you stop before you enroll all of your planned subjects. And on average, you need fewer subjects, but sometimes you need all of the planned subjects, and that's a larger number than if you didn't have the interim analyses in the first place. So it works really well if you have the efficacy you expect, and it provides a nice hedge if the efficacy is a bit less than you expected. This general concept has been modified to create the seamless phase 2/3 studies. These studies start with multiple doses, have one interim analysis where you decide which is the best dose, and then you finish up with that dose and the control arm. It's well controlled, and you can continue the subjects in your best arm across the phase 2 and phase 3, and then combine all of that data together. So you get your first phase 3 study for fewer subjects. There's additional savings in not having that downtime between phase 2 and phase 3 that we always have. The challenge is in knowing enough about your intervention that you can design your phase 3 before you've started your phase 2. And that's a tall order for most new compounds. These studies are appealing because of the potential for large time and money savings, but really only work in a situation where you know everything except the dose. Now, the pruning design is an adaptation of the group sequential that occupies the other end of the spectrum, when you know hardly anything about the intervention going into phase 2, and you need to do a lot of learn before you get to confirm. In these designs, you start with a range of doses and then do a lot of interim analyses to get rid of the arms that aren't as desirable as you go. In this case, desirability means efficacious, safe, easy to take, all of those things. The payoff for these designs is that you get to look at a lot more doses for the same number of subjects as a traditional phase 2 study, but you still get a lot of subjects in the doses with the highest potential. Now, the downside is that they're not well controlled, so we can't use these as a pivotal study in any way. Other, more traditional adaptive designs may not get the big headlines, but they can be just as useful in their own way. The adaptive randomization designs, for example, are widely used and do not cause nearly as much controversy. These designs look at the characteristics of a subject and then decide which treatment to randomize the subject to based on what the previous subjects with those characteristics have been randomized to. The adaptive piece here is therefore the randomization status. We make decisions on future randomizations based on what we've already randomized. The purpose is to keep the treatment arms balanced across the factors that might affect subjects' response to therapy. Now, this eliminates bias when we're estimating the treatment effect at the end of the study. We can be more sure that the effect we're seeing is due to the intervention and not big differences in, say, age between the two treatment groups. Sample size recalculation designs serve a completely different purpose. These studies have really gotten a bad reputation in recent years, and honestly, some of it is well-deserved. From the FDA's perspective, increasing the sample size partway through your study, just because you don't see the original treatment effect you planned, is pretty much tantamount to cheating. The basic premise here is that you do an interim analysis and increase the sample size if the assumptions of your original protocol were wrong. And there are two different types. There's blinded and unblinded. Now, the blinded version looks at pooled data, like rate of events or variances. The unblinded actually looks at all of those, plus the treatment effect. As you can imagine, the blinded is much more acceptable in terms of controlling your type 1 error, since you're not even looking at your efficacy effect or testing any hypotheses. Statisticians often claim that if you are unsure of your assumptions going into a study, you should do a group sequential study. That design is much more powerful than recalculating your sample size in the middle of your study. However, exactly how endpoints are going to turn out is much harder to predict than, say, next year's fashion. So even if you feel good about the placebo rate or the variance estimate that you choose at the beginning of the study, there's a lot that can go wrong, and you might be really off, which is where the sample size recalculations come in. The problem is when this method is used to save studies that were designed without forethought, and that's where the bad reputation comes in. Used cautiously, however, these designs can be yet another useful tool in your toolbox. So what have we learned in our adaptive designs introduction? 1, no matter what kind of adaptive design you're doing, the critical thing is to identify what aspect of the trial you're going to review and then make changes based on that review. 2, make sure that the design you're using is going to give you what you need and not just be trendy.